Okay, well, good evening, everybody. It's my pleasure to welcome you to an evening to talk about honey. And I've traveled all over the world, and in my travels, I think I'd be hard pressed to find a country as a whole that does a better job of marketing honey than New Zealand. And we have the pleasure tonight of having three experts on honey in New Zealand. Terry Braggins, who will be our first speaker, and is going to talk about MＡNUKA. Maureen, how do we properly pronounce your last name?
Maureen. She's not hearing you.
Conquer is what it looks like, but I don't know if that's right. Frank?
It's Maureen.
No, the last name.
Oh, she's, oh, golly.
All right, so the other expert we have here who be the moderator is Frank Lindsay, and he comes at it from the practical side. He's a commercial beekeeper. Wow. So you will notice that Terry is not the person we announced for the talk tonight. His colleague John had a conflict that came up, so Terry was good enough to step in to represent the MANUKA industry, and I'm a scientist, so I'm glad to see Terry here because he knows the chemistry of MANUKA inside and out and so on, and that's kind of the cornerstone of this unique monocultural honey. So with that, I think we're already 2 minutes past the hour. So Terry, you have the forum.
Thank you very much, Jerry, and hello everyone, and thanks for the opportunity to talk to you from the bottom of the world, and winter as well. So it's great to have the opportunity. As Jerry said, that I've stepped in on behalf of John Rawcliffe, who is the coordinator of the Unique MANUKA Factor Honey Association of New Zealand, and I've compiled these slides. John is more marketing related to the MANUKA honey, and Maureen's going to talk about that, whereas I'm more coming from the science point of view, but I'll lead in with some interesting statistics about the volume of honey being exported and the value. So very briefly, the MANUKA Honey Association is an incorporated society. It now has over 140 companies as members, so that beekeepers, exporters, marketers, and they pay a license fee, which helps to fund the work they do. There's also a quality mark, the UMF quality mark, which is a grade, and it's based on the biopotency or the antibacterial properties of the honey, which I'll talk about in a minute. It represents over 75% of all New Zealand MANUKA honey retail exporters, and it also runs a program of product authenticity assurance, so chemical characterization that I'll talk about today, MANUKA classifications and market verification. So MΑNUKA, which is a Maori name, so the indigenous population of New Zealand, call that, which is the Leptospermum scoparium plant. New Zealand has just one variety, Leptospermum scoparium. There are many other varieties around the world. It's quite a diverse plant. If you look at it from various parts of New Zealand, it can be quite small or quite large. And the picture there on the right shows the flowers in full bloom, and a great photo in there shows the nectar flow coming out of one of the flowers. It's a perennial species, and as I said, found throughout New Zealand. The MANUKA is heavily laden with cultural and traditional association pertaining to the local Maori population. And it is, MANUKA is not a generic word for the species Leptospermum or scoparium, and it's specific to that plant, and it's not to be used for other scopariums, for example, from Australia. So to be MANUKA, it has to come from Leptospermum scoparium specifically, but also has to come from New Zealand. Now, here is 2 graphs of the relative price per kilogram of Manuka honey, starting from about 2010 and up to 2017. I was unable to have time to update the information until 2020 because of the short notice I had, but the trend is pretty much the same. So you can see the price, the low price to the high price for Manuka honey ranges from about $10 right up to, in 2016, over $140 per kilogram for the high value stuff. Whereas you look at the other honeys around the world, or in New Zealand, sorry, they remain quite low. So Manuka honey does have a premium. And here are the exports by most countries around the world of honey. And you can see China up until, again, 2017, about 110,000 metric tons. You can see New Zealand is quite a lot, 8,500 metric tons. However, when you look at the relative value of the exports from New Zealand, you can see that although China has about, I think, 15 times more exported honey, the actual value of the product is roughly the same. So what it's meaning that New Zealand honey commands about 15 times more value per kilogram than other honeys. And you can see that the figures around about $275 million. Well, in 2020, that's now up to 426 million. So it's still growing very quickly and strongly. And this is just another way of expressing the same thing. So on average, in 2011, that the New Zealand honey was getting about $38 a kg, and from China about $250. And we all know the various ways that China produces very low cost, low value, supposedly honey. So what is the difference between Manuka honey and other honeys? Why does Manuka honey command such a high price? Well, as you know, most honeys, if not all, have some sort of bioactivity or bacterial, antibacterial activity, and mainly due to peroxide value. So there's hydrogen peroxide produced from glucose using glucose oxidase enzyme. Hydrogen peroxide is detrimental to the viability of bacteria, so it will kill it, amongst other things, osmotic pressure and pH of the honey. But the hydrogen peroxide, when the honey is used as a topical treatment on wounds, if there is any hint of blood around or red blood cells, then that peroxide activity is neutralized because the red blood cells contain catalase, which is an enzyme that breaks down the hydrogen peroxide. So Dr. Peter Molan, he's now passed away, but he was the person in New Zealand who mainly discovered an extra activity of New Zealand Manuka honey that was unique. And this is called the non-peroxide activity. So it's independent of the peroxide activity, and it remains active if it comes across blood cells and topical wounds. It's been subsequently shown that the active ingredient is methyl glyoxyl, or MgO. And it's formed in the honey in the hive. It's not present in the nectar, whereas the dihydroxyacetone, which is the precursor of the MgO, is found in the nectar. And this is what happens with Manuka honey if it has been stored for a period of time. So the conversion of the MgO to, of the DHA to MgO over time is time and temperature dependent. And the top graph shows the rates of decline of DHA and the increase in MgO at 20 degrees, and the lower graph shows the same reactions occurring at 27 degrees. So it can occur quickly. Now, it's not as though 100% of the DHA converts to 100% MgO. there is some loss, and it's a combination of the DHA breaking down to something else, and also that the MGO is breaking down at the same time. So once the engine is depleting, and the DHA gets down to about the same concentration of MGO, you'll end up seeing the MGO starting to decline. And this is important information. The consequence of storing honey for a long time is that there are other indicators of freshness of honey that deteriorate. And one of them is HMF. And so HMF production is also time and temperature related. So HMF, the increase in HMF and also the decrease of enzymes such as diacetase, which are used to see if there is temperature abuse of the honey, they also change in MＡNUKA honey that is not temperature abused, because you get the same effect if you store the honey at a high temperature for a short period of time than as you do if you store a long period of time. So all other honeys other than MANUKA around the world tend to be sold as free flow through as soon as possible and get rid of their inventory before the next season. Whereas in MANUKA honey, it's stored for a long time, perhaps 2 years in some cases, and you will inevitably get these storage-related changes. What we've done in analytical laboratories is that we've been able to model this change, and what we, from these calculations, we can actually predict or forecast what any honey's MGO, DHA and HMF levels will be at any point in time at any temperature. And we use this now for all our customers who get this free calculation when they get the tests done at our laboratory. And so they can work out how long they can keep the honey before it's past its use-by date, and what the potential will be. So not all MANUKA honeys are equal in terms of the levels of MGO or NPA or UMF. There are some lower ones, for example, starting at about a 5, and some really high ones are 20 plus. So by using this sort of prediction, we can help the determine when to sell their honey. I referred to the non-peroxide activity, and this is determined by a bacterial test in an agar plate, but we can also work that out by a correlation with the actual methyl glyoxyl concentration, as this is done quite easily by HPLC in the laboratory. And the lower graph or table, you can see that you can get quite a range of values. The average for DHA about 840, MGO about 110 on fresh honey, and of course that will change over time. And leptospermum is another marker which I'll talk about soon. So historically there was an issue that there were so many different activity labels and criteria used for a lot of producers in New Zealand, and some of them were called total activity, so that was a combination of non-peroxide and peroxide, or just NPA non-peroxide activity. There was the unique MＡNUKA factor activity, just activity, the Molin gold activity, and methyl glyoxyl percent pollen, and even a K factor, which has been used. And you can see some of the honeys that I've got in front here showing the different types. And it confused the consumer overseas, so it was decided there needed to be something more discerning and unique that could identify what a good quality MANUKA honey was. And typically, overseas and in international markets, there are a number of tests to identify a monofloral honey, and they range from pollen, colour and conductivity, and of course now the chemical profiles. The interesting thing with the traditional methods for New Zealand honeys, at least, is that the pollen, colour and conductivity are inadequate in distinguishing MANUKA honey from other floral types. And so the graph down on the bottom left shows a number of different New Zealand introduced and native plant-based honeys. And you can see the orange MANUKA identified in the ranges. So it would be hard to differentiate based on colour with a value of, say, greater than 73, that it was either a Kānuka, Manuka, Rata, Thyme, Rewa Rewa, Ling, and Honeydew. So there's no distinction there. Similarly, on the right with the conductivity, again, the Manuka is very similar to a lot of the other honeys. So it didn't really offer us that degree of separation. Also, the pollen, I'm not a believer of pollen as an indicator of floral type, other than perhaps a forensic opportunity. But if you're looking at shape, there are two graphs here, or two pictures, electron microscope pictures of pollen. One of them is a Kānuka, which is very similar to a Manuka, and the other one is Manuka. So to most people, it's very hard to tell the difference, so we can never distinguish that. So what we decided is to set out to look for chemical markers. And of course, there's a circular argument. And to get the right sort of marker, we need a library of authentic monofloral honeys to determine what tests can be used to determine authentic monofloral honey. So you go around and round, and mindful that the bees can travel a difference, and there's competing plants for nectar flow at the same time, and that changes within season, for example, Manuka and Kānuka, that it gets quite hard to distinguish. And also there are regional and seasonal variations. So we needed to characterize all the honeys in New Zealand, and our strategy was to first look at the nectars. So we collected a lot of nectars from a lot of different floral types from all over New Zealand over 2 seasons, and we developed the methods in analytical laboratories. And as you know, there's a small amount of nectar, and we perfected methods to be able to fingerprint and identify unique chemicals in the nectar. We went on the basis that if we only found it in the nectar and found it in the honey, then that honey must come from that nectar plant. And so that way we developed these fingerprints and profiles of what we believed were authentic honey. So the UMFHA started that project in about 2014-15, and then the following year the MPI, that's the Ministry of Primary Industries, a government organisation in New Zealand, decided to run an independent program. As it turned out, they independently took the samples from both nectar and from honeys, but they sent the samples to analytical laboratories as well. The only difference between the two is that the UMFHA took hives and placed them in known areas of monofloral plants, whereas the MPI project mainly relied on beekeepers to supply them with what they thought were monofloral honeys. The MPI also introduced a pollen DNA test, which is quite specific, going on the basis of that if it hasn't got the Manuka pollen DNA in it, then it must not be Manuka. We've subsequently found that it's not a very strong test for proving Manuka. We know now that that storage time of Manuka, that the DNA is degraded and it's no longer detectable in high-grade honeys. And also the levels to be positive are set so low that nearly every honey in New Zealand tests positive to the Manuka DNA. So just to reiterate, we're selecting unique chemical markers common to nectar and honey, and we did it for over 600 samples for the UMFHA, but about 700 for MPI, and it was by HPLC, high resolution mass spectroscopy, and looking for unique markers that were higher abundant, reasonably easy to detect, and were stable over long-term storage of the honey. So that was an untargeted approach, and the picture down the bottom shows an example of all the compounds that you may find in honey, and it may well be many of them are common to all floral types, and it might be only a few that are specific to one of the monofloral honeys. And indeed we found that, and from that, with the MPI program, you can see we picked out those chemical markers the 2-methoxyacetophenone, the hydroxyphenolactic acid, 3-phenolactic acid, and 2-methoxybenzoic acid. And then the UMFHA, moving to the targeted one, the methylglyoxylin DHA, leptospermum, leptyridine, and again the 2-methoxyacetophenone. UMFHA has also applied for a provisional patent for about almost 20 other unique markers which are undergoing some more research. This is how we went about it. So if you look on the left, there's a graph with the different floral types showing the concentration of DHA so the top left one is a nectar. And then if you look down at the DHA in the honeys from those floral types, you can see it's present in there. So therefore, it must be from that plant. Similarly, on the right, the leptospermum is the nectar leptospermum. And again, you see leptospermum in far more abundance at the bottom right-hand graph, which is for the honey. But you will also see that there is some leptospermum in the other honeys as well. And the reason for New Zealand being a small, quite diverse floral country is that you will get crossover and contamination between floral types. But when we just look at the nectars, it's definitely not present in the other floral types. The Ministry of Primary Industries have now come up with a definition, this is 3 years old, of what MΑNUKA is. And it's in three categories really, the monofloral MANUKA and then the multifloral MANUKA, so a mixture. And the third one is non-MANUKA, of course. So the 3 PLA, the one on the top, for being monofloral has to be above 400 and above 5 for 2-MAP and above 1 for the other 2 chemicals. And hidden there is that the pollen DNA has to be less than 36. It's an inverse thing, so the higher the value, the less DNA. And then the multifloral for the 3 PLA has to be between 20, above 20 but below 400, and the 2-MAP has to be greater than 1 and less than 5. So that's what's used now in New Zealand. So it has to qualify like that to get a label of MANUKA honey on the jar before it's exported. The MPI won't let it out. And so it's a good way of controlling the quality of the honey. In addition to having to have those chemical markers, they stipulate now that any label of honey cannot have a health or a therapeutic claim. So you can't say it's got a total activity or a non-peroxide activity. But the UMFHA were able to convince the organisation that if they used the UMF grade, so it's a quality grade rather than saying it's a health or therapeutic claim, they were able to keep that on the label. And you can also use the methylglyoxyl because it is actually constituent and represents and can be expressed as a concentration. So the chemical markers are crucial for quality control. So crucial for authenticity of genuine floral honey type. So if it's got to have certain concentrations of chemicals for it to be authentic, and the idea is to prevent any adulteration or mixing with other honeys. It is also used to harvest the best quality raw material, and you can see that by the various grades, UMF grades, that the higher grades have the higher bioactivity. Obviously, post-harvest handling is important for quality and the assessment of the final product when it goes out on retail. Obviously, chemical analysis is very important for detecting harmful toxins and pesticides, and also for any fraudulent practices. Are countries just getting sugar syrups and adding some flavourings and colourings and adding a few enzymes and then calling it honey? And also gives customer assurance so that they know they're getting true to label. It says it's MANUKA, it's of a certain quality, and they can be confident of that in the future. So future challenges and where we're moving is we're looking, and MPI is looking at constant improvement. Does the definition fit the purpose and cover the natural variability? We're finding that there is one or two of those chemicals seem to vary around the country quite a bit. It may well be, for example, up in Northland, that some companies up there are disadvantaged because their good Manuka honey is probably not being graded because of the 2-MAP concentration may be set too high. We're also developing a curated reference collection of New Zealand honeys, and this is underway at the moment. So in addition to finding the chemical markers for Manuka, we have also discovered a lot of chemical markers for the other New Zealand monofloral honeys. So the idea is that when we have this library, it will be used as a reference collection for researchers to develop new methods, and also a good reference for when we start to do more using other techniques such as NMR. The NMR you may have heard of is becoming a new way of testing the authenticity of samples in their collection. So if you put the wrong samples in there, you'll get the wrong result. So what the part of this library or this reference collection is to have an authentic collection of samples that can be used to reference in the NMR. We're also interested in harmonizing methods. There are new methods coming out around the world, particularly related to sugar syrup adulteration that are replacing the AOAC method. And we're keen to be participating in harmonization of that. There are some changes that occur in Manuka honey over time, as I said earlier, and one of them is that there's an apparent C4 sugar increase. So in the good Manuka honeys, if you get a C4 sugar value of say 3 and you store the honey for 1 to 2 years, the level will end up at about 8 or 9. And it's not because C4 sugar has been added, but because one of the components in the test, the protein component, seems to shift over time. And there's a lot of research done. We know it happens, we've proved it happens, but we're not quite sure why it's happening. I have an idea, but we just need to do more research in that area. So it's internationally, the world needs to know that there's not only is Manuka honey unique from the point of view of being bioactive, but also it's unique, and it's one of the few, if the only, honeys that are matured like a good red wine over time. And there will be some subtle chemical changes, and unfortunately those changes have been picked up by people doing certain tests. And so we need to educate the world that these changes do happen. We're also, finally, there's a large program going on to protect the name Manuka. It, as Maureen will talk about soon, we've spent a lot of years and a lot of money building up the market brand for Manuka. And the last thing we want is Me Too organisations and countries and companies coming on board and exploiting our commercial uniqueness and the honey Manuka, which must come from New Zealand.
5 minutes, Terry.
I've finished. John commissioned this photo to be taken. It's a beautiful one of what the bees would see in the nectar coming out from the flower. So thank you very much for listening, and I'm most willing to take any questions.
Well, thank you, Terry. It was very interesting. And I've learned some things that I didn't, I followed the chemistry a little bit and so on, but I've learned quite a bit more today than I knew. Our moderator for tonight is Frank Lindsay. Almost anybody who keeps bees in New Zealand knows who Frank is. And then people who belong to the Eastern Apicultural Society have probably seen him several times at EAS. But Frank's a commercial beekeeper, and I've asked Frank to address the questions. If you have questions, please, there's a few of them listed on the Q&A. Address your questions, type them into the Q&A, not the chat. So Frank, go ahead.
Okay, the first question, we're using a lot of acronyms, and I think some of them are confusing. MPI is our Ministry of Primary Industries. The other ones I just put down as NMR. People may not know about nuclear resonance testing, which the Americans are now developing to detect fraud. And the whole world's really accepting that. They're starting to accept that way it's going because it detects fraud honey better for C3s and C4 sugars. Is Manuka honey more beneficial honey bees than other types of honey? That's an unusual one. Terry probably couldn't answer that. You're a chemist, but I don't. It's high mineral content, but the bees certainly winter well in New Zealand on it, and like any other honey. What other tree plants outside New Zealand have the ability to produce high levels of MGO? Do you know that, Terry?
None known. There has been a lot of research in a lot of countries to come up with a Me Too product from another plant, and I am unaware of any that would produce the levels of MGA, well, specifically the levels of DHA in the flowers to produce the MGO. So that's unique. In the Manuka honey, the molan, to be very effective of topical treatments of wounds that have been very hard to heal. So it may be ulcers that people have had for a number of years, or some of the bacteria that are flesh eating that normal antibiotics have been unable to cure. And there's plenty, if you just Google online, and there's some pretty horrific looking photos of people with very ugly looking wounds before and after treatment with Manuka honey. And so that's where it all started, in that medicinal area. And it's really followed over the perception that it must be good for you also when consumed.
Terry, can you explain a little bit how DHA works and what it really means? Because people live in the beekeeping in America have probably never even heard of it. Okay,
so DHA, or dihydroxyacetone, is a chemical that's involved in metabolism in the body and in plants, and it has to do with the glucose cycle. And it's unknown at this stage why it's so prolific in Manuka nectar, as opposed to a very similar plant, Manuka nectar. And even in Australia, there are some species of Leptospermum that don't have it at all. So we don't know why it's there, but the conversion of the dihydroxyacetone to methyl glyoxal, although they sound different names, chemically the only difference between them is a water molecule. And so one is active and one is inactive. So it's there as a precursor or a store. We don't know why. It ends up in the honey and it's converted by a chemical reaction into MGO. That's all we know.
Do you know the difference between Manuka and Manuka? Or is that...
Yeah, up until about 1984, is that Manuka was called Leptospermum ericoides, and the botanical scientists decided that it was actually a different genus, and so they renamed it Kunzia ericoides instead of Leptospermum. And they're quite similar to look at, and the flowers are similar. They grow in similar areas around the country. And the only way I can tell the difference is when I clutch the branches, is that the Manuka's leaves are soft and the Manuka's leaves are prickly and prick my hand. So when we look at the chemical profiling of all the honeys in New Zealand, the honey with the most chemicals in there, these polyphenolics and other compounds, is Manuka, and it's closely followed by Manuka. And as we go through the New Zealand floral types, there are fewer and fewer of these chemicals are found in there, almost to the point when we get to an introduced species, clover, that there are very few of these chemicals in there. So at some stage, the Manuka has been very close to the evolutionary processes, MＡNUKA, but they have diverged over time.
Yeah, there's just as a beekeeper, just looking at it, you can look at the difference between MANUKA. MANUKA flowers, develops flowers right through all the branches, whereas CANUKA only flowers on this year's growth or last year's growth. The flowers are slightly smaller in CANUKA, and really it's been developed. There's not much nectar in it because it's developed along with our native bees, and the native bees are the main foragers of the pollen and a little bit of nectar. They don't require much, so it doesn't really produce something. So you've got to have 3 fine days at 16 degrees at nights before it actually secretes a bit of nectar that the bees will actually take on. So that was one of those. Does crystallization of MANUKA affect the MGO content?
Not that I am aware. We at the laboratory test all types of honey, tens of thousands of honey samples a year, and we get young, fresh honey, so that's straight out of the hive, or just in a storage drum, so before creaming or after creaming, and our predictions predict accurately what it should come out at, and retesting it, it's fairly close.
There's probably a question here we'll bring up with Maureen, and that is, so he's travelled New Zealand 2019, tasted several brands of MANUKA and the several different taste profiles. And that can be beekeepers determining whether it's MANUKA or not, because locally it's not really, people don't test it to the same degree. Taste profiles, that's again, doesn't matter about poor spelling. What's the reason the tea tree flower produces so much DHA? Well, you've just explained that. What forms of delivery MANUKA honey? Does one just slap the stuff on a spore spot? Right. Well, that's that's medical grade.
I think the most sales of MANUKA honey go through for consumption. The volumes that go to medical are a lot lower, and there's quite a stringent criteria for using MANUKA honey on wounds. It has to be certified, particularly in America, by the FDA, and it's actually certified as a medical, medical instrument, or rather than being a drug. So it has to be, I think they make sure that it has low bacterial count or fungal count, and I think it has to be sterilized in some way.
Yeah, the last question here is, how consistent from season to season is the chemical makeup of the MANUKA honey, or does drought and rainy conditions affect it?
Well, there are seasonal differences affect the growth, the timing of the flowering, whether or not there's nectar flowing when it's flowering, and so therefore a lot of that is expressed through the chemicals. It won't actually change the type of chemicals present, but there is changes in the concentrations of those chemicals in the honey. And it also, another factor is, there's very few honeys around that you can put your hand on your heart and say 100% MANUKA honey. And it's because of the diversity of floral types in New Zealand, and the desire of the bee, and what it wants and where it wants to go and get it at any particular time, and what is available will end up determining what concentrations of the chemicals are in the honey. And so that is why MPI set, rather than setting levels that it must be, for example, between 5 and 7, or etc., is that there are minimum levels set to allow for this variation. We just have to remember honey is a biological material, and it's subject to all those variants, and somehow we have to put a label on it for our consumers, but it's inevitable that there will be variation.
And just like with normal beekeeping, we have droughts, we have winds, and if it rains, you don't get any. And we've had seasons where very little produced, but luckily New Zealand, basically in the North Island, has 5 different distinctive areas, and beekeepers move from the Northland down to the Coromandel, and then down the East Cape and down to the Wairarapa, and then back into Taranaki and then inland. So there's, as you go inland, it's higher up in the plateau, and therefore it flowers a little bit later. So there's a slight overlap, and beekeepers take advantage. So if they don't get it in one area, they can get it in others if they're lucky. Well, that's really all the questions. Thank you, Terry.
Oh, there's just one question at the end. I see how many laboratories, or which laboratories outside of New Zealand test for MGO. In America, I think there is Eurofins down in New Orleans do the test. There are some laboratories in Europe, Eurofins, Intertech, and QSI in Germany. And then there is FERA in York in England, I know do it, and I'm sure some other laboratories. You can always, if you want some honey tested, you can always send it to me at Analytica. We do have a import license and quarantine facility for bringing in honey samples into New Zealand for that purpose, and we get a lot from Australia in particular. But New Zealand does not import honey commercially, and so it has to be under a certain specific license that's audited and under quarantine.
And just for your comment, what's your 3-in-A test now cost? 3-in-1 test?
Oh, I think it's down in the high 20s, $28. It depends on the volume you put through. It was very interesting exercise before we got the THA and HMF was $300, and we came and combined it into one test, and now it's down into the 20s of dollars. So it's been a big saving for the industry.
Yeah.
Well, I have to comment that in my visits to New Zealand and so on, and when your prices are good on Manuka, we're known in the States for semi trucks carrying loads of bees from one coast to the other, but I have never seen beekeepers in the States flying beehives into the mountains using helicopters. So.
Yeah, that's great to see. That's great to see.
Sometimes the break even is about 7 kilos per hive, so it doesn't take much to make it pay.
Well, that is interesting. I do have a question. The way Zoom sets up on this panelist attendee webinar, I can't ask questions through Q&A, so I'll ask it. I guess it's because I have a microphone. So we see occasionally here in the States some debates about, is Manuka a healthy thing for a person to eat, or is Manuka possibly, with all the chemistry in it and so on, less than the best honey? Is there any downsides to consumption of Manuka? And I get this on the internet, so I have no idea how to answer that question.
The interesting thing, I'm unaware of any clinical trials that look at what happens to Manuka honey on consumption. There are a lot of ideas. There's been a lot of work done about the topical treatment, but when it comes to the clinical trials for human consumption, as you appreciate, and as the pharmaceutical companies definitely know, it's extremely expensive to run clinical trials in the tens to hundreds of millions of dollars. And I don't think there is anyone prepared to fork out that sort of money from the New Zealand industry. But perhaps Maureen can talk more about perceptions and marketing of honey, and would know more than me about that.
Sydney University's done some study, and they found that people eating honey, it gets right down into your bottom of your bowel and helps the bacteria. So that's one of the things, that it's not destroyed by you through your initial stomach, but it actually feeds the bacteria in your bowel. So all honeys would do that, I presume.
Yeah, it's interesting. I just saw one last question. I'll quickly go. Someone wanted to know, explain again the difference of hydrogen peroxide in Manuka and non-Manuka honeys. So the hydrogen peroxide is produced in all honeys, but less so in Manuka, because one of the reasons is that it seems to be broken down by various reactions related to MGO. But MGO is not found in other honeys. And so it has the same sort of antibacterial activity, but it has got nothing to do with peroxide, whereas the other honeys have peroxide in them.
And that's how Dr. Molan found it, by killing the peroxide. He found this honey still kept killing bacteria on a plate.
Yeah, yeah.